this video we will discuss about the biochemical pathways that helps in the mechanotransduction in general, and then we will discuss this topic with an example of Alzheimer disease. So in this what are the different ways by which biochemical pathways are being disturbed or can get affected using mechanotransduction force. So we will start with the steady state conditions where this piezo is the piezo helps in helps in transfer of these ions, and that results in changing these mechanical forces into the electrochemical stimuli. So in the general case or in the healthy normal case, it is closed, piezo is closed. However, because of the extracellular matrix, that is membrane tension, shear stress, or ECM composition, that leads to the stretch of this piezo, and that enables the transfer of the ions inside the cells, or the Ca2 influx increases. That will leads to the different change in the signaling pathways, such as AKT pathways get triggered through the phosphorylation. That leads to a trigger of this mTOR pathway, and the targeted genes can get affected, and also this YAPTAZ pathway can also be affected. So the other way is that it helps in this next way by which this mechanotransduction pathways can be affected is by formation of this collagen 4 in the extracellular matrix. It will also result in the opening of this piezo, and it will cause the similar effects that we have observed in the step 2, or that we have observed in this second criteria. And along with that, what it will does is it will also affect this WNT pathways, and this beta catenin destruction complex get inhibited, and all of them will cause epigenetic changes or modulation of the target genes. The next pathway is about this, that when this piezo gets open, then it will also affect the cytoskeletons using this affectin or this affectin filamin A. Or when the rod-like structures of affectin filaments are formed, then cells are less prone to move and more prone to cell adhesions. And it will inhibit, and these affectins, once these affectins rod-like structures are formed, they will inhibit the closure of this piezo, thereby the influx of this Ca2 or other ions will be increased as these piezos are not being able to closed because of the rod-like structures formations of these affectin filaments. These are the different ways by which biochemical pathways can cause mechanotransductions. So now let us quickly go through the examples. So another example, that is how the mechanosensing biochemical pathways works in this Alzheimer disease. So because of the change in the ECM, the extracellular matrix, because we know that Alzheimer disease are caused because of the amyloid formations. So the amyloid will help in the change in the ECM matrix, or change in the ECM axis, or it will cause ECM degradations. It will affect the ECM that is surrounding the cells, and at the same time, or because of this, the phosphorylation of this NFTs will be increased within the cells because of the mechanotransduction forces that occurs due to the change in the ECM's degradations around the cells. The next part is about this amyloid, is about the these amyloids will help in this opening of this piezo, because of this the calcium ions can get entered into the cells, and this entering of the calcium ions will cause the inhibition of the pro-inflammatory response. So the immune response will get suppressed as the pro-inflammatory cytokines get reduced, and Also, these amyloid plaque formations interact with the cell surface membrane or cell surface transmembrane that will lead in the down regulations of this 5AP that we have discussed in our previous videos. And also, this interacted with these integrins to form down regulations. The integrins interact with the changed extracellular matrix compositions to cause down regulation of this firm T2, and because of the upregulations of this MIR 4502, it will increase the risk for Alzheimer disease. At the same time, this L1 cam cause the A beta 42 clearance. It also promotes the Alzheimer diseases. And these YAP down regulations will also cause this NFT formations, phosphorylated NFT formations, that is also caused by ECM degradations or change in the compositions of the ECM in the part A. Next part is about this filaments rod-like structures formations, and that usually occurs because of the excessive inflammatory cytokine release. So this effect is completely opposing of this. This effect is slight some kind of opposite of the part B, in which excessive inflammatory cytokine release will cause these filament rod-like structure formations, and these filament rod-like structure formations in other way will cause the permanent opening of this piezo that results in this more Ca2 plus ions enters into the cells and affect other functions, such as they will cause MAP2 activation of kinase cascades, such as MAP2 signaling pathways get activated, and this more of this NFT phosphorylated NFT formations. And the last part is about this about the effect on this nucleus. So these rod-like structures, filaments formations will cause decreased chromatin mobility. So the chromatins will become more rigid. At the same time, it will also affect the nucleus, as the nucleus will become more rigid because of the filaments rod-like structure formations will restrict the free movement of the nucleus and the other components, intracellular components within the cells. So the chromatin mobility, because of that, the chromatin mobility is also goes down, and this laminin A C formations increase that further aggravate the stiffness of this nucleus that is present in the cells that are affected with the Alzheimer disease. So this is just an example using Alzheimer diseases.